Picking up where we left off, we started talking about the structures that are going to be found within inside the cell. We first start taking a look at the endomembrane system. What that really incorporates is that all the different internal bound organelles that are actually membrane bound. So things like the endoplasmic reticulum, Golgi apparatus or Golgi body, lysosomes, peroxisomes, vacuoles, vesicles, all of those actually are surrounded by a phospholipid bilayer. Now, what their job is to do is they do a key role in creating and or breaking macromolecules. Now, in our last chapter, we talked about how the basic monomers of either proteins or lipids are put together by condensation reactions, but we also talked about how we can break them by use of hydrolysis or adding water. Now, if you can see here in this lower right-hand side, what this is showing you is an image of the structure of a smooth endoplasmic reticulum. And then production of it is going to move its way into the Golgi body. But we're going to see that here in just a second. So if we look at the ER, what the ER is a factory system that it manufactures membranes, and it is the job of performing building functions. Now, the two different types of ERs we do have are the rough and smooth. Rough has a ribosome, so its job is to not only create proteins, but also to transport those proteins. Where the smooth does not have the ribosomes, and its job is to help create those phospholipid bilayers. So if I'm looking at the actual structures here, I can see the transport vesicle that is going to be created and pinched off from this is your ER. Okay. Now notice the location where the ER is. It is right directly next to that of the nuclear envelope. So the messages from our genetic material that's going to come right out of the nucleus is going to directly into that of the ERs. And then from there, the ER is going to pinch whatever it had been created and transport it out into a vesicle. Now, factory processing, what's going to take place here is the building and breaking of materials. As I already said, in the rough ER, it's going to be doing that for proteins, but in the smooth ER, the functions here is for creation and breaking down of other things. Enzymes that are going to be found in the smooth ER are going to help create lipids, oils, phospholipids, steroids. These can be things like cholesterol and or sex hormones from androgens or estrogen. The breakdown of the glycogen in our liver, as well as from the hydrolysis reaction of that to create our glucose. And also can be utilized to detoxify drugs. Rough ER, you can see in this picture is a good picture to study, to know, because what we want you to be able to synthesize and tell me is how does the nucleus, the genetic material inside here, actually work its way to embed proteins in the plasma membrane. Ultimately, if I'm walking my way through it, if I zoom in there and I can see the structure, I am having the nucleus material in here. That material is going to leave through these pores in the nuclear envelope, land on a ribosome, lands on the ribosome, creates the protein, and it's going to be transported through there. Once that protein is created, it then is pinched off of the rough ER. And from there, it's going to then be shipped out to the Golgi body, as you can see on the left-hand side here. So it's being shipped, and it's coming in the open door of the Golgi body, which we call the cis face. So it enters the cis face of the Golgi body, and it gets deposited in the Golgi body, where it's then going to be assembled and packaged into a particular location so it can identify where it needs to go. From there, it goes to the medial, so that's middle of it, and then eventually it goes out the trans face. So if you look down here, down on the bottom there, it shows the trans Golgi body location right there. So it pops out the trans and eventually will work its way through until it reaches the plasma membrane. Now, the thing is, it doesn't just float here. It's actually going to be moved by motor proteins along microtubules, but that is going to be in a later PowerPoint. You'll see that microtubule development. Okay? Bring that back down. So synthesis membranes, we're going to see that in manufacturing systems. They're going to build new membranes as the ER, where the synthesis of proteins is important. So we definitely need the endoplasmic reticulums. Here's another illustration showing you that of the smooth and rough ER, smooth and rough ER located again on our systems. Showing that of our plant also have the smooth and rough ER, because not only do they need to create phospholipids, but they are also going to create the structures of proteins. Okay, what's a function? of the Golgi body. Function is to finish, sort, and ship materials. So its job is to transport the proteins and lipids that were just created, maybe enzymes that the protein was set up to be, hormone, whatever it may be. It's going to be working its way out of the cell, or it even can be utilized in the cell. So as I mentioned during the last couple seconds ago, we're looking at the structure of it from the cis to trans face. Flat membranous sacs where the cristae, cisternae is located. It enters through the cis face and it exits through the trans face. Okay. So I believe that is also one of your questions that's on your study guide or your testing your understanding, is looking at that structure. So again, make sure you can map that out from the nucleus all the way to the plasma membrane. Lysosomes, another fluid-filled sac, phospholipid bilayer, and inside there you're going to have enzymes that were created by the smooth ER that are going to be embedded, as well as the rough ER, I should say, that are going to be embedded into the sac, and that sac will float around inside that of the cell, and it's going to be used to help break down other foreign debris. Now, what is their job? Their job is to eat. We call them the cleanup crew. They call them the little stomach. Their job is to help break down solid materials that are entering into the cell that shouldn't be there. So here's another illustration of cellular digestion. Lysosomes fuse with food molecules, so as the food comes in comes this digested sac that contains enzymes. That sac will then start breaking it down. Okay, another illustration showing you that same thing here. So as the food comes in, it mixes with that food particle, breaks it down, so eventually all that digestion is going to be taking place. Lysosomal enzymes work best at a pH of 5, so if we start to change the pH levels, we can actually start to break down lysosomal functioning. So if the functioning of the lysosomes don't start to work, we can end up starting to have major issues taking place, and chemicals or foreign debris could start playing an effect on the structure of the cell. Things that could happen like that is an example of Tay-Sachs disease. So if lysosome digestion enzyme doesn't function correctly, what it would have been from a protein, or maybe the enzyme that was supposed to be developed was not created. Now, if you remember from our last chapter, enzymes are proteins. So if I changed one particular letter in my mRNA sequence that was supposed to correspond to my DNA or my protein sequence, my amino acid sequence, that ultimately changed the shape of the protein, which then no longer allowed to do its particular function. Now, in this case, it doesn't digest a fat tissue that starts to deposit, and this lysosomal storage disease are usually fatal. So we start developing a whole bunch of fat on the brain, and the child usually dies around the age of 5. That's a horrible thing, but ultimately came down from one particular amino acid, but that one amino acid that was changed was due to one nucleotide that was changed. I believe it was changed from a C, a cytosine, to a T, a thymine. Sometimes it's supposed to work that way. You know, one of the things that the mitochondria can do, and that's things that we've seen, is this process called apoptosis. Apoptosis is programmed cell death, so it's a critical role in the programmed destruction of cells. Its job is, if a cell is going and it's growing too fast, it's not doing its normal functioning, the mitochondria can go up and basically put the self-destruct button on. Unfortunately, cancer has found its way to override that. Now, other fluid-filled sacs that we do have, phospholipid bilayers, are peroxisomes. They have other digestive enzymes in it, and their job is to help detoxify and help break down chemicals that start to develop in our body. For example, what they do is produce peroxide, hydrogen peroxide, but we need to break hydrogen peroxide down into water. Now, a lab that we're going to be doing in a week or so is the use of catalase. Catalase is an enzyme, ends in ASE, that is used in our liver to help break down hydrogen peroxide into water and oxygen gas. So we're going to be seeing this process taking place. But these peroxisomes are in both animal and plant cells, but they break down these fatty acids to sugars. But in that process of doing it, you start to create peroxide. We just can't have that. That's a toxin in our body. So with that sense, it's nice that it can convert it into energy. However, we need to be able to make sure we can get rid of that wasted byproduct so we have other enzymes, like catalase, to break that down. Here's showing you an example of the function again. It's very similar. Food vacuoles go in, so the cell eating of the vacuole/vesicle. It is more of a storage site. Sometimes be fluid filled, and then if it's fluid filled, it can then be used to break things down. If the fluid is containing, maybe it contain water, but maybe it also contain enzymes of other sorts. You can have a contractile vacuole, which I mentioned the other day, is found in protists. It can help expel water out of their body. And or central vacuoles we found in plant cells that are going to help increase the turgor pressure that's going to be found with inside plants to allow them to stay upright. What's their function of a vacuole is to store. Whether it's storing proteins, storing byproducts, storing pigments, compounds. It's selective membrane, selectively permeable, so it does control what goes in and out because it is a phospholipid bilayer. So reviewing the endomembrane system, all the membrane-bound sacs that goes through, nuclear envelope is a membrane-bound system, the ERs are membrane-bound, the vesicles are membrane-bound, the Golgi bodies are membrane-bound, the plasma membrane is membrane-bound. So all of those are surrounded by those phospholipid bilayers. Okay? Questions on that? If you do have questions, please jot those down in your binders or your notes, and we will come back to it as soon as we get back into class. Thank you.